Hello, welcome to 13 Universal. Learning beyond barriers. This is cell structure part 3, subject content, and this is schedule. So the question is, what is a cell? A cell is the basic or fundamental unit of life, basic or fundamental structural and functional unit of life. Okay, so these are cells. You can see why this is a plant cell, this is an animal cell, this is a bacterial cell. These two are eukaryotes. Why? They have well-defined nuclear membrane enclosing their DNA. Okay, this one has no nuclear DNA, it just has cytoplasmic DNA lying, all right, in nuclei. Okay, let's go. So look at this. This is endosymbiont theory. Okay, now it states that mitochondria and chloroplasts were bacteria. So these guys were bacteria. What happened to them? That now live inside larger cells of animals and plants, which is why chloroplasts and mitochondria have circular DNA. So this endosymbiont saying that mitochondria and chloroplasts, they evolved from bacteria. What is the evidence? Both mitochondria, chloroplasts, and bacteria, they all have circular DNA. And it's called endosymbiont. That mitochondria and chloroplasts decided to partner to live inside animal cells and plant cells. Okay, that's endo, inside animal. And symbiont means mutualistic. Okay, um, why mitochondria provided energy, chloroplasts made food, the animal cells and plant cells sheltered these guys, and everybody was fine. After some time, they lost something. There's something that bacteria have they don't have: cell wall. So these guys lost cell wall and became dependent on animal and plant cells. See, this day they still live inside plants and animals. Okay, that's the theory. Now look at it. Let's go to ribosome. So ribosomes are non-membrane bound spherical organelles. Non-membrane, they lack membrane, and they're the smallest organelle. Okay, what do? Site at which mRNA transcribed from the nucleus. All right, this is where mRNA is formed in the nucleus by transcription during protein synthesis. The first stage of protein synthesis is transcription. DNA is used as a section to copy genetic information into mRNA. mRNA is called messenger. Messenger mRNA carries the genetic information from the nucleus to where ribosomes, and then the information is then used to make polypeptides, and that is called translation. Okay, so translation occurs in ribosome. Ribosome is a site for translation, site for protein synthesis. With the help of tRNA, therefore help with protein synthesis. They are membrane bound. I think I mentioned it. Of course, there are two components of ribosomes: ribosomal RNA and protein. Ribosomal protein. So ribosome contains nucleic acid called ribosomal RNA. There are two types of ribosome: 80S ribosome and 70S. So 80S ribosomes, where are they found? They are found in eukaryotic cells, cells that have well-defined nuclear membranes. Okay, that means that 80S ribosomes are found in the cytoplasm, scattered in the cytoplasm of eukaryote, or attached to RER on eukaryote. Is that okay? Then we have 70S ribosomes. So the 70S ribosomes, okay, very important. They are found in chloroplasts, mitochondria, and bacteria. So they are not found. They are found in these chloroplasts, mitochondria, and bacteria. Their size is small, 18 nanometers to 20. 80S is 22 nanometers, 25. So this is bigger than this, right? And then ribosomes also have something else. They have two subunits: small subunit and large subunit. So the small subunit is a site where mRNA attaches to, binds to, for translations occur. Large subunit holds everything in position: mRNA, tRNA, so that amino acids can be joined together to form polypeptide. Guys, let's go. So this is the first check. Let's check this, guys. The diagram shows functions of four cell structures: W, X, Y, and Z. So look at them. All right. So these are the functions. So W makes glycoprotein, and of course transport protein, Z, Y makes lipid, X makes protein. Which row correctly matches the cell structure letter representing a function? So remember, you are supposed to pause this video and just try the question. Okay, this is knowledge check 1. All right, Golgi body, guys. Golgi body. What's the function? Golgi makes glycolipid or glycoprotein, so that's W. So ribosome makes protein, that's X. You can see rough endoplasmic reticulum transport protein made by ribosome, Z. Simple. And then Y makes lipid, that's smooth endoplasmic reticulum is a site for synthesis and transport of lipids. So the answer is A. Let's go. So you see the ribosome, I was telling you, this is a large subunit, guys. Okay, this is a small subunit. So you see messenger RNA attaches to the small subunit, exposing the genetic code, which is called codons, so that the tRNA transporting amino acid attaching it to the growing chain, okay, so that peptide bond can be formed. See, peptide bonds have been formed connecting this amino acid to form polypeptide. This is a growing chain of polypeptides, okay? All right, so let's go to lysosome. So you see the size, 0.1 to 0.5 micrometers. So don't forget that lysosomes is continuous with Golgi. Golgi forms lysosome, right? So a single membrane bound with no internal structures in animal cells. Now, what do? They contain digestive enzymes, hydrolytic enzymes. They contain potent hydrolytic enzymes. Okay, these enzymes break down large molecules. For example, they contain protease enzymes, which breaks what we call peptide bond in proteins. Okay, then have nuclease enzyme that breaks down the phosphodiester bond in nucleic acids. They also have lipase enzyme breaks ester bonds in lipids, and they have carbohydrate enzymes which breaks down glycosidic bond. Is that okay? So now then that is kept separate from cells to prevent damage. So you see that what is their function? They are responsible for intracellular and extracellular digestion. Okay, so they break down wanted structures, example old organelles in the cells or whole cells. In phagocytes, they digest bacteria. All right, that's about lysosome. Okay, the lysosome is just spherical. All right, just like this, guys, like this. It's large vesicles produced by no. All right, large vesicle made by Golgi. Okay, look at it. So you just have right there inside our enzymes. So microtubules. So microtubules are very important. What are they? Long hollow tubes. Now, long rigid hollow tubes that make up the cytoskeleton, which helps to determine cell shape. So one of the things they do is that they are long hollow. Remember, we'll talk about structure, we'll talk about function. Long hollow tubes that make up the cytoskeleton. This cytoskeleton helps to determine the cell shape, but they don't just help to determine the cell shape, they also help to move, all right, organelles, vesicles, and of course molecules. So look at them. So this is the microtubules made up of alpha and beta tubulins. So the tubulin proteins, tubulins are proteins. So tubulins are they are the monomers, the subunit of microtubules. So you see that microtubules are made up of proteins. Anything that is made up of microtubule will contain this protein. Okay. For example, microtubules form central. So if you are asked what is the biological molecule found in central, what do you call it? Tubulin. Okay, that contains dimer joined end to end to form protofilament. You can see all of that. They form protein, which round, okay, fold from round this inner core, right, to form a cylindrical with a hollow center. So forms an intracellular transport. So that intracellular support system by moving along secretive vesicles, that's cytoskeleton. Okay, organelles are moved by this intracellular transport system. Okay, cell component on its surface, they move it using ATP. All right, so that is what I'm trying to say. This is one of the things that make up microtubule. Centrosome, a pair of centroles at right angles that is involved in nuclear division and act as microtubule organizing centers, MTOCs. Okay, so this is central. All right, so central is formed by 9 triplets. Don't forget it, 9 triplets of microtubules. Microtubules extend from central, so they radiate from central and attach themselves to the kinetic core of chromosomes, forming spindle fiber. Centrols duplicate and they pair of centrols, right? They move to opposite poles. That's at the beginning of cell nuclear division, okay, of the cell. 2 centrose regions, thus separating sister chromatids during nuclear division, okay. So you can see that 2 of them will migrate to the opposite, the duplicate and migrate to the opposite pole. Let's see this. So you can see that. So during nuclear division, can you see? This is the poles made up, found central here, central. So these centrals, what do? They radiate microtubules. These are microtubules, this long cylindrical hollow. Okay, they radiate from centrals. That's why centrals are called microtubule organizing centers. Okay, so when these microtubules are arranged, they form what we call spindle fibers. So what is the role of that? Now at metaphase, okay, of mitosis, all right, the spindle attaches to the kinetic core of the chromosomes and then align themselves along the equator. But you see, this is anaphase. At anaphase, these spindles or the microtubules start shortening, all right, at the poles. When they shorten at the poles, you know what they do? They contract and they pull the sister chromatids apart, all right. They pull them apart, all right, towards the opposite pole. So that's what they do. They separate the sister chromatids. Now at telophase, what they do is that they push the 2 daughter nuclei further apart. So you see that centrols at the basis of cilia. So centrols also are found at the basis of cilia and flagella. So they form the basal bodies and they act as MTOCs. So microtubules extended from basal bodies into cilia and flagella help with their beating movement. All right, look at what I'm trying to say. So let's check it out. So look at this. I think we've done this. Okay. It said 4 students were asked to match the function with appearance of some cell structures in an animal cell. So let's try this. Okay. Let's try this. The functions were listed by the number. So A, synthesis of polypeptides. B, synthesis of lipid. 3, packaging of hydrolytic enzyme that will remain in the cell. Then the appearance were listed by the letter. Now these are the appearance, the structure of these organelles. So V says membranes which surround and enclose inner cavity. What does this speak to? This speaks to endoplasmic reticulum, either rough or smooth. Okay. All right. They have membranes which surround, right, and enclose inner cavity. Now W says non-membrane bound spherical structure. This speaks to ribosome. This is ribosome. Okay. Okay, so let's do it. Okay, this is ER. Okay, whether rough or smooth. Okay, this is ribosome. Good. Ribosome. Okay, yeah, that's non-membrane bound spherical. Good. Ribosome. Sorry. And this one that X nuclear pulse, that's talking about nuclear membrane, nucleus. That talks about nucleus, NOC. Okay, Y talks about non-membrane bound cylindrical structure. That's central. Okay, so that's central, guys. Yeah, right. Flattened, Z talks about membrane bound flattened sacs. That is Golgi, right? Golgi. Good. Golgi. Let's look at how you go. Golgi apparatus. So that number 1 here talks about synthesis of polypeptide. So which of them synthesizes polypeptide? Okay, so either rough endoplasmic reticulum or what? Or ribosome. So let's go with ribosome. So ribosome is number 1. Okay, the second one says, synthesis of lipids. So rough, that will be smooth endoplasmic reticulum. So it will be represented by this synthesizes lipids, so that means that goes with 2. Okay, 2. And then the third one says packaging of hydrolytic enzymes. The only thing that does is Golgi, so Golgi goes with 3. So you can see, guys, we have found our answer quickly. So let's go. You see that 1 is what? V. No, there's 1. Okay, this is 1. Yeah, 1 is W. Okay, W. 1 is synthesis of polypeptide, and that is done by ribosome. What is the structure of ribosome? Non-membrane bound spherical. That's ribosome. Okay. All right. So A and B chosen, C and D cancelled. So let's go to number 2. 2 is synthesis of lipids. What synthesizes lipids? Smooth endoplasmic reticulum. V. Okay. So B eliminated now. The third one is packaging of hydrolytic enzymes that will remain in the cell. Packaging is done by Golgi. So 3. So the answer is alpha. Okay, guys, look at what I'm trying to tell you. This is microtubule. Remember that microtubules can be found as part of central and then it can also be found as part of flagellum and cilia. So in cilia, they form a sphere of duplets. So duplets of microtubules. Duplex means 2. 9 duplets. 1, 2, 3, 4, 5, 6, 7, 8, 9, with 2 singlet at the center. So anytime you see this, that talks about the arrangement of microtubules in flagellum and cilia. All right, look at it here. That's extended. 9 duplets, okay, at the circumference, and then 2 singlet at the middle. But when we're talking about central, so central is made up of course, at the base members. That's central. C central is now triplets. Okay, this is a triplet. 1, 2, 3 triplet. But it's going to be 9 triplets. 1, 2, 3, 4, 5, 6, 7, 8, 9. It doesn't have a singlet at the middle, so don't forget that. Okay, so look at this cell. This is a plant cell, 40 micrometers. That's huge, guys. See, this is okay. In plant cells, vacuoles at the center. Okay, of course, don't forget that nucleus is at the periphery. Okay, yeah. So vacuole at the large central nucleus, peripheral nucleus, large central vacuole, peripheral nucleus. The nuclear membrane is extended, is continuous with this rough endoplasmic reticulum. So nucleus is the largest, okay, followed by chloroplast. So chloroplast at the edge, push the edge by the vacuole. This must be a mesophyll cell because they synthesize a lot of food. Okay, they have large number of chloroplasts. Can you see? Now you see mitochondria is smaller than chloroplast. Chloroplast is maybe about 10 micrometers close to nucleus, but this is like 1 micrometer or thereabout. See Golgi. All right, so now see this is plasmodious matter, cytoplasmic link. So let's go, guys. So yeah, look at this. The electron micrograph shows a structure found in cytoplasm of an animal cell. This is cytoplasm of an animal cell. Okay, it says which biological molecules are found in this structure. This is structure. Which biological molecule? Now, which structure is this? Which organ is this? Pause this video and try it. Now, if you check, we have how many? This is a triplet of microtubules. Let's count it. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 triplets. That must be a central. This is a central. And central is made up of 9 triplets of microtubules. Microtubules are made up of tubulin proteins. So tubulin proteins are the only biological molecule here. So the answer is 2 only. Is that okay? All right, look at this. Chloroplast 5 to 10 micrometers. Okay. Now this is cell structure is found in plant cells in the palisade mesophyll mainly because they make food. Spongy mesophyll, okay, surface which carries out photosynthesis and ATP synthesis. Guys, don't forget that chloroplast has similar structures like mitochondria. First of all, it has 2 membranes, envelope, okay. Also, it has 70S ribosome and circular DNA. You can see it has internal membrane system. All right, this internal membrane system is called grana. Grana consists of a stack of thylakoid. The thylakoid contains chlorophyll. That's a site for light dependent stage. Then we have the stroma, site for light independent stage. You can see it has double membrane and contains flattened sacs known as thylakoid. Chlorophyll is embedded in thylakoid membranes. Thylakoid stacks on top of each other to form grana. Grana are linked by lamella. These structures are present in the matrix called stroma. Contains starch grains, circular. You see, they have circular DNA and 70S ribosomes. So there are 4 things or 5 things it has in common with mitochondria. Of course, number 1, they have circular DNA. Number 2, they have 70S ribosome. Number 3, they have double membrane. Number 4, they have internal membrane. Number 5, they all make ATP. Right. So guys, look at this. There's 2 different types of cells, P and Q. These are 2 different types of cells, P and Q. We are broken up by using ultrasound and their contents analyzed. Both types of cells contain smaller circular DNA. So smaller circular DNA, that means that they have chloroplast and mitochondria. But guys, pause this video and work it out. Let me see how you're doing. Okay, so circular DNA, right? The 2 cells are said, so they must have chloroplast or mitochondria. But let's go. The circular DNA from P all carry the same base sequence. That tells us that P has one organelle, either mitochondrion or chloroplast, because it carries the circular DNA carries the same base sequence. But those from Q were of those types, were of two types with different base sequences. So that means that Q will have either chloroplast or mitochondria. Let's go. P here. Heart muscles is an animal cell, has mitochondria, no chloroplast. Root cortical cells, these are plant cells, but they don't have chloroplast. They have only mitochondria. So one mitochondria. This is supposed to have mitochondria and something else. Okay, so it's not the answer. Lymphocytes, white blood cells, animal cell has mitochondria. Mature red blood cell, animal cells has mitochondria. But they say contaminated by bacteria anyway. But you know, it has mitochondria, even though this has circular DNA, right? It has circular DNA, but you see, this is another cell. The circular DNA is inside this bacteria cell. We're talking about this Q, sorry. Okay, the Q, of course, you can see, should have 2 types, so mitochondria and chloroplast. So mature red blood cells, of course, has mitochondria. Phloem tube have mitochondria, so this cell should have 2. Root cortical cells, mitochondria. This leaf mesophyll cells, wow, mesophyll cells make a lot of food, so they have a lot of chloroplast and also have mitochondria. So our answer is delta. Okay, which row describes the membrane surrounding each cell structure? Answer it. Chloroplast, post video answer it, has double membrane. Mitochondria, double membrane. Nucleus, double membrane. So the answer is delta. D. Cell wall, 10 nanometers rigid as it contains fibers of cellulose polysaccharide. So in plant cell, it's made up of cellulose polysaccharide. In animal cell, it doesn't exist. In proctists, it's also made of cellulose. Okay, but in fungi, it's made up of chitin. In proctists, okay, in bacteria, it's made up of peptidoglycan. So it gives the cell its definite shape, prevent it from bursting by osmosis, allowing turgidity, may be reinforced by lignin for extra strength. So cell wall in plants has lignin. It has cellulose, but it can also have lignin there. Also have hemicellulose. Okay, it can also have pectates. So there are other substances apart from cellulose. Lignin is there, hemicellulose, and pectate. All right. Freely permeable. Now, this is another one. Plasmodium matter, pore-like structures found in cell walls that allow a link. So 2 things. It links the neighboring, the cytoplasm between 2 cells. Find through the cytoplasm. Okay. So this guy has 3 functions. Acts as a link. Number 1, helps in communication. Number 2, helps in transport between 2 cells. Large vacuole and tonoplast. Now, tonoplast is the membrane of vacuoles. Surrounded by partially permeable tonoplast, has cells, okay, consists of enzymes. You see, the vacuole has enzymes, sugars, waste product, pigment, mineral salt, oxygen, CO2, Okay, they're all in there. All right, so this is plasmodial matter. It connects the cytoplasm of this cell and that cytoplasm of that cell. Okay, do you get? So it can help to transport. So first of all, it joins, links them. It allows transport, allows communication. That's cell wall. This is cell membrane here. Okay. So eukaryotic organisms with a true nucleus and have membrane bound. That this is nucleus. They have membrane bound organelles. Okay. And of course, plants, fungi, protozoes, animal cells, they're all eukaryotes. Prokaryotes, they lack nucleus and they have simpler structures, bacteria. So look at this, guys. So now this is bacteria. Okay, this is circular DNA, cytoplasmic DNA lying freely, okay, in nucleus at a region called nucleus. They have plasmid, okay, of course, peptidoglycan cell wall. They have 70S ribosome. Do you get? They have this infolding called, of course, this infolding. They have capsule, flagellum, right? Now these are, the structures. But viruses, guys, viruses are, they belong to another classification, their own. All viruses are parasitic, okay, because they lack cellular organelles. All right, so they reproduce by infecting and taking over living cells. They infect host cells and then hijack. Look at it. The virus DNA RNA hijacks the protein synthesizing machinery of the host cells. So they hijack the RNA DNA polymerase to make their own DNA. They hijack ribosome. They make their own protein, which helps them to make new viral protein. So they are strict intracellular parasites. So things about viruses: number one, they lack cellular organelles. Number two, they actually they are endoparasites. Okay, the endoparasite. Okay, so they hijack the cellular organelles. Okay, and they have capsule. No, not capsid. Capsid. We're going to see that. Let's go to the next page. But before that, let's answer this question. The diagram shows 3 cycles: 1, 3, and 5, representing chloroplasts, mitochondria, and typical prokaryotes. Okay, so which rule correctly identifies the 3 cycles and some of the structures that are shared between them? So I want to know whatever that is in cycle 1 and cycle 3 have 2 in common. Whatever is in cycle 3 and cycle 5 have 4 in common. Okay, so cycle 1 is chloroplast. Cycle 3 is mitochondria. Do they have circular DNA together? Yes. Yeah, mitochondria and prokaryote, do they have 80S ribosome? No, they have 70S. Okay, so A is wrong. B, chloroplast and mitochondria, they don't have 80S, so B is wrong. Prokaryotes and mitochondrion, they have circular DNA. Good. Mitochondrion and chloroplast, they have circular. So C is correct. Now, why is it wrong? Prokaryotes and chloroplast, they have 70S. That's correct. But chloroplast and mitochondrion, they don't have 80S. They have 70S. So answer is C. Right. So look at it. It said it is possible for a bacterium to synthesize a eukaryotic protein. Okay, because it has ribosome. Okay, this bacterium has 70S ribosome, so it can make protein. This involves introducing a eukaryotic gene into the bacterial DNA. The eukaryotic gene is then translated by the ribosome. Okay, in the ribosome by the bacterium. What explains why bacteria cell can produce a eukaryotic protein but cannot produce a eukaryotic glycoprotein? It can make protein, but it cannot make glycoprotein. Okay. Now A says bacteria do not have rough endoplasmic reticulum. Well, the question is, do you know the answer? Pause the video and try it. Guys, watch. Don't forget that the bacteria, okay, this bacteria can make protein, but it cannot make glycoprotein. So the answer, the question, the $1 million question is, what organelle makes glycoprotein? What organelle? Of course, Golgi. That answers our question. Golgi. This bacteria doesn't have Golgi. Okay, so it can make glycoprotein. It can make protein because it has 70S ribosome. Okay, and DNA, but it cannot make glyco. It cannot add glucose to protein. It can modify it. All right, guys. Which structures are found in typical prokaryotic cells and also in typical plant cells? Prokaryotic and plant cells. Now, cell wall is found in, okay, pause the video and solve it. Cell wall is found in 2 of them. because prokaryotes have peptidoglycan cell wall. Okay, this has cellulose cell wall, but you see histone, telomere histone, they are not found in prokaryotes because histone, right, helps to fold, to wind DNA, right? And then most prokaryotes don't have a histone protein. Okay, they have only DNA. So because they don't have histone, they can have telomeres. Tonoplast, all right, is found only in plant cell. Bacterial cells don't have tonoplast. They don't have large that vacuole. They lack membrane-bound organelle, and tonoplast, they are the membrane of vacuoles. Okay, so the answer is A. All right. Which role shows a comparison that is not correct between a typical prokaryotic cell and a typical eukaryotic cell, plant cell? It's not correct. It's not correct. Okay, let's check this. DNA, prokaryote DNA not associated with histone. Yeah, correct. DNA associated with histone, correct. This is correct. No endoplasmic reticulum present. Yeah, they don't have membrane-bound organelles. Endoplasmic reticulum present. Yeah, this is correct. Peptidoglycan cell wall. Yeah. This has cellulose. This has peptidoglycan. This is correct. So all ribosomes are approximately 18 nanometer. That's 70s ribosome. All ribosomes are approximately 22 nanometers in diameter. Well, this measurement is for 80s ribosome, but don't forget that eukaryote also has chloroplast and mitochondria, okay, which have 70s, which is also 18 nanometer. So this should be all. This should be. It has both ribosomes with that are 18 nanometer and 22 nanometers in diameter. So D is not correct. Guys, this is a dangerous guy virus. All viruses have, they lack organelles, they lack cell organelles, but they have something. They have protein coat. This protein coat is called capsid, right? Containing a genetic material. This genetic material can either be what, DNA or RNA. Is either DNA or RNA. It can be both. Okay? And don't forget, guys, that for DNA, watch this, guys. DNA has how many bases? It has adenine. Okay, remember? Adenine, you have guanine, you have cytosine, you have thymine. RNA has what? It has what? Adenine, it has guanine, it has cytosine, it has uracil. So a virus will always have these three bases, okay? Because they are found in both DNA or RNA. Remember that the sugar in DNA is deoxyribose. So a virus can either have deoxyribose or ribose, which is RNA. Okay. So let's look at the next one. So it's a which feature shown in diagram can be present in viruses? Viruses have protein coat, yes. They have RNA, they have DNA, but because they say viruses, but if they say a virus, that means either one of these. But viruses, some of them have DNA, some of them have RNA, but they don't have organelles. They don't have 70s ribosome. Okay, so it's 412. Is that okay? Or 124. That's B. All right, you solve this question. What is present in all viruses, all prokaryotes and all eukaryotes? All viruses have proteins, protein coat. Pause the video and solve it. All viruses have protein and they have, yeah, genetic material. Now, if you say ribose, ribose is found only in RNA virus. Deoxyribose is found in DNA virus. Thymine is found in DNA virus, but cytosine can be found in both RNA and DNA. So C is the answer to that question. Which polymers are present in all viruses and all prokaryotes? All viruses have polynucleotides, DNA or RNA, which are also found in prokaryotes. All viruses have capsid protein, capsid, which is polypeptide, but not all of them have polysaccharides. So the answer is 1 and 2, guys. Don't forget this. B, right? Which role could be correct for a virus? I want to pause the video and answer it. All right, for a virus doesn't have to have carbohydrates. It will have DNA. It must have protein, either A or B. Okay, DNA or RNA. So if it has protein, A or D, okay, check DNA. A is wrong because it cannot have DNA and RNA, but it can have DNA If it has DNA, it cannot have RNA. So D is the answer. So guys, you can see these are the differences between prokaryote and eukaryote. Take time and read it. So you see the average diameter. See the size of prokaryote is 0.15 to 5 micrometers. This is 10 to 100 micrometers. Okay, 1,000 to 10,000 times volume bigger. DNA circular, lies prevalent. Okay, cytoplasm is naked. The guy is naked. It's not surrounded by nucleus. Linear DNA, circular DNA, surrounded by nucleus associated with histone protein forming chromosome. This doesn't form chromosome. Okay, ribosome 70. This has both 80 and 70. ER absent. All membrane-bound organelles are absent. Cell wall here is peptidoglycan or murine. Okay, this is cellulose for plants, chitin for fungi. Right, cell division, binary fission. For eukaryotes, mitosis or meiosis. Transcription occurs, okay, simultaneously, consecutively. All right, guys, look at it. Which range would include most prokaryotic cells? Okay, remember we said from 0.5 micrometers to 5 micrometers. Now, remember when we did, pause the video and try it. When we did our microscope, okay, if you are moving from micrometers, all right, to what? To what is called nanometers. You multiply by 1,000 times 1,000, okay, times 1,000. All right, so if you are going back, divide. So this is, divide this one by 1,000. It will give us 0. Okay, 0. Okay, 0. I think 1, which is less than 0.5. So I think it should be from 100 nanometer, which is 0.1 micrometers. Okay, between that, right? But that's still low. Yeah, but this is between this 1 to no, this will not work. Okay, because this is 0.1. Okay, what I'm trying to say is that, look at it, 1 nanometer is 2, and A is between this and that, but B is between 0.5 micrometers to something before 100. So B is the answer. C is between this is 100 micrometers square, far bigger than the size of a prokaryote. So the answer is B. Okay, the size of prokaryote is between 0.5 micrometers to about 5 micrometers. Okay, so of course D is way bigger. So answer is Bravo. All right, which structures are found in palisade mesophyll cells and photosynthetic prokaryotes? Okay, yeah, so all cells have cell membranes will be there. Cellulose is found only in plant cell. Ribosomes is found in both. Chloroplast is found only in plants, so it's only 1 and 3. Cell surface membrane and 3 will be found in 2 of them. So 1 and 3, which is C. All right. So some scientists think that mitochondria evolved from bacteria that entered the cytoplasm of a different cells and were able to survive there. Which structural feature of mitochondria support this mitochondria hypothesis? So I want you to try and answer this question. All right, remember that the only structure that will support is structures that are found in both prokaryotes and mitochondria. Folded internal membrane is not found in prokaryote, but it's found in mitochondria. So A, B, C can be the answer. So it's the circular DNA is found in mitochondria. 70S is also found in both mitochondria. So circular DNA found in both mitochondria and prokaryote. Same for 70S ribosomes found in both mitochondria and prokaryote. So the answer is delta. Okay, so it says some of the cell structures are listed. You can see that. Which cell structures can be seen in electron micrograph? This electron micrograph. So nucleolus. Pause the video and try. We can see this nucleus. You can see that darkly stained. This is a nucleus. You see the nucleus, the nuclear membrane. Okay, we can see the nucleolus stained. Chloroplast, we can't see chloroplast here. Microtubules, we can see it here. Ribosomes, yes, we can see it. Dot. So 1 can be seen, 4 can be seen. Mitochondria, yeah, this is mitochondria. This is it. Okay, this is not chloroplast. Plasmodium matter. Yeah, this is a link. Can you see this link here between this cell and the next cell? So we can see nucleolus, ribosomes, mitochondria, plasmodium matter. So 1, 4, 5, 6. 1, 4, 5, and 6. D. Thank you guys for holding forth. See you in the next. Please remember the next video. Remember to like, subscribe, okay, share your experience with us. This is Shedro. See you later. Bye. Bye bye.